So this is the link for the feedback form. Many of you have filled it up, many of you haven't. So I would kindly request you to fill this up as soon as possible. Okay, so today's let us begin right now, so today we are talking about RNA-seq processing and some amount of differential gene expression. So we are going to look at the gene expression today, and gene expression study is commonly called RNA-seq and are divided into 4 main steps. They are pre-processing, alignment, quantification, and differential gene expression. For pre-processing, we will be using a section of our platform which takes raw reads with sequences from raw reads, and then we remove the technical artifacts or variation which are not relevant to the biological perspective that we are going to look at. In terms of alignment, we are going to align the read to the reference genome. And now the reference genome can be well annotated. It has a GTF file, and we will tell you about what GTF file is. And we will also look at non-annotated genome or non-model organisms that do not have GTF file, and therefore the different types of data is going to change. The type of the data in this case would be different. Quantification. Quantification is ultimately what we want to get is a count, how many transcripts of a specific gene are present in a sample, given the reads are very short, 30 to 300 nucleotides long, and the genes could be very long, thousands of nucleotides, and you can see how variations within the sequences of a single gene and differences between different exons and their expression can play a role in the quantification. And finally, when we have the data, what can be done with the data so that we look at the statistical significance and probability of how we can compare 2 conditions or a number of samples of a group so that we can get differentially expressed genes using p-value, using false discovery rate, and fold change. Okay, so let us go into how we have generated our data, the data that we are going to look at. So we have a FASTQ file. What does a FASTQ file have? A FASTQ file have the sequence. As you can see that it has the length. For example, the length of this sequence is 36. These are short reads, and they also have quality scores. These are the quality scores of this particular so starting from here to up till here is the entire description of this particular read. So it tells you about the length, it tells you the actual sequence, and also it tells you about the score or the quality of that file, of the particular sequence. And we also have FASTA file, which don't have any quality score, but we only have the different elements, which are the component nucleotides of the sequence. We also have something called GTF file, gene transfer format file. The GTF file reports the beginning and the end of the genome element. We will look at the GTF file in a few moments from now. For example, this could be exons. These are exons actually that belongs to the certain genes, so that the information is recorded here in these files. So moving on, this is what are we studying actually? When we are looking at the RNA-seq expression, what are we actually studying? We are studying the RNA transcription. So we are studying RNA transcription. Some amount of structure of RNA is also very important. So we will discuss accordingly. The transcribed macromolecule out here is RNA, which obviously we all know that it comes from the DNA. And the DNA produces mRNA, which produces proteins. Here we can have 2 types of RNA. We can have, we can have the total RNA, and also we can have mRNA, which for library preparation. Now the transcription have some story inside, and the process is a little complex. So on the right of the screen, you can see the entire procedure of the transcription process. The DNA produces complementary RNA chains, and the only difference between the DNA and the RNA is the uracil base. So the thymine is replaced by the uracil nucleotide and are added at the 3' end. And the RNA has a poly-A tail, which is cleaved later on. Thereafter, capping occurs. So capping occurs at both the 3' and the 5' end of the RNA. And for the RNA, and this capping is very important because it protects the RNA from denaturation when it leaves the nucleus into the cytosol. Thereafter, alternative splicing occurs, where the introns are removed from the RNA strand. And now we only have the exons. Here. And different exons could be combined in different combinations, giving rise to multiple transcripts for a single gene. Okay, now see that you have a gene here, and you have the GDF file, which contains the information of this gene. And then you have these reads, in the form of a FASTQ file. As you can see, the genes are represented by the yellow and the pink and the blue and the green and so on and so forth. And the intermediary regions which are grayed out are basically the introns. So the green, pink, and blue combine to form. So if we click on, we can say that, see, the green, the pink, and the blue combines together to form isoform. This is something isoform 1, and this is isoform 2. So by combining the reads, the isoforms are formed. And this is important to understand because later on we will be identifying these isoforms when we are trying to analyze the RNA-seq data. And then you have combined all of these isoforms to understand the quantification of this. And this will be all done based on the GTF file so a GTF file which we have originally I was telling you that the GTF file might be, some genomes are annotated, some genomes are unannotated, so they're non-model and model organisms. So the GTF file is very important out here. So this is how a GTF file looks like. It tells you about the chromosome. It talks about the exons and also the starting positions of the exon, the ending position of the exon. So it tells you about the exon IDs, the gene IDs, the transcript IDs, and the exon IDs, and all these sorts. So this is ideally GTF file, what it looks like. Okay, so from the technical perspective, you have let's say you have gene A, gene B, and you have gene C. Now gene A and gene B produces the transcript, and they are now they are shattered. They are shattered into reads. They are broken down into small reads. Now the process is it starts with the first step is shattering, followed by the adapters ligation, and then which is followed by at the beginning and the beginning of the sequence and the ending of the sequence are being capped by the adapters. Then we go for the amplification of these sequences so that more copies are produced, so that we have multiple copies, and then there is the chances of reading those reads, the chances of making the mistakes in reading those reads are less. And then we do the reading. We actually read into the thing. So we add the adapters, identify the beginning and the end of the file, and then we do a PCR amplification. And so you have enough quantity of reads at the end of the day. So shattering, adapters ligation, PCR amplification, and reading. So therefore, before since we do this adapters ligation and PCR amplification, we start with pre-processing. What is pre-processing? In pre-processing, we remove the adapters and the PCR duplicates and statistical analysis of typical coverage, so that we remove the exact replicates which looks suspicious and eliminate them accordingly. And then we map them into the known transcript or on the genome, and this this is entirely done to find a new transcript as well. Also, we use a combined strategy to find both, both the gene. We try to find novel transcripts, or we identify the known transcripts as well, and then we quantify. We quantify out here, and then here I would like to take a pause and definitely ask you to log on to the server, because the next step is actually going into the server. So I will take a pause here and ask you to log on to the server if you haven't. So there seems to be some questions out here. Okay, so many messages. Okay, show Mudeep how to join. You have already joined, I guess. When I go to my dashboard, it asks me to register for the course, which I have already done. We will look into it, Sunita. Okay, so I guess all of you have been able to log on to the server. If you haven't, shall I wait for a few moments? So let me go to the server now. So okay, this is the server. If you have logged on, you will be seeing this this thing here. And let us go to the front page, which you would see when you log on. So there are 2-3 pipelines, demo pipelines, which we have already uploaded into the server, so that about you first kind of go through a guided tour of the entire process, and then try to do your own analysis. The first pipeline that we are going to run, you can find this small arrow out here, and then you click on the demo pipeline. So as we already said, that this is an AI guided platform. So the immediate next step would be highlighted. So I click start, I go to OK, and then you can see many of the immediate buttons that that are there. So if I go to the the immediate slide that is important, this is the pipeline that I'm going to build right now. So it, as you can see, it starts with pre-processing, and then I directly align the reads onto the transcriptome using bowtie2t, and then I derive the table of expressions, which is our purpose, and then we end. And this is, we call it a simplest pipeline, simplest RNA-seq pipeline. Why do we call it a simplest pipeline? Because it is not looking at the read alignment and to the genome, but just looking at the known transcript without any pre-processing. That is also important in the process of RNA-seq data analysis. So let us build that pipeline, starting with Trigonometric. And there are educational pop-ups which you can study, and there are mentions, or there are specific publications mentioned out here which you can read. So that is the whole reason why we have built this demo pipeline, so that you can understand. Go to PCR clean, click OK, and the immediate next step is bowtie2t. Okay, and then you do the ArcSym table of expressions, and you go click OK, and then of course you end like that. If you don't, I mean, this is how it ends. And all these things are covered in our transcriptomics 1, transcriptomics 2, and transcriptomics 3 courses. So this is the simplest pipeline that we have right now in front.
Excuse me, here one question, please.
Yeah, Sunita, yes, please go.
Yeah, here, is this the only pipeline we can use, like top hat and this? Do we have HiSat and string tie, those things also here?
Yeah, of course, because as I was telling you, this is a demo pipeline, right? So where the input files which are required for our purpose is already uploaded. So The first thing is that you have to kind of understand that what kind of files have to be uploaded onto the server. That is why we have created this demo. And also, this sort of pipeline takes quite some time to learn, some time to run. Let's say 1.5 days or 2.5 days or something like that. So before actually going into the actual ocean to swim, it is better to learn swimming in a pond. So this is why we have done this demo pipeline, so that you know that the basics of the swimming, and then go into the bigger picture and a bigger waters or deep waters to really take the swim. Does it answer your question?
So, so if I know this and if I want to do another pipeline, then I go to what? What do I do?
We'll show you. We'll show you, Sunita.
Okay.
Can you just
All
hold
right.
on?
Okay.
Because I can understand your question. I know that you have specific
yeah, that's the main reason I want to go one-on-one. We'll talk later.
Sure. And one more thing is that this is what I think, these are some of the basics we are covering. So if you can go to them,
right.
it would be.
No worries.
Thank you very much. Take care. Okay, so this is a simple pipeline that we have built. So there are more complex stuff, as Sunita was telling. So we can create some other type of pipeline as well. So if I right now go to the the presentation, if you see that, let's say this is the GTF file that you can see on the right, and let's say these are your reads. So any read which is aligned onto the GTF file, this kind of reads make more sense rather than these which does not match to any of the, it actually falls on the gap. These read also make a little more sense because this is mapped to GTF. Let's say if it is GTF1 and this is GTF2, these two elements are mapped like that. But this particular thing which is falling on the junction, falling on the gap, does not really make sense. So this is what we try to identify. So there are several steps. It's not like that you build the pipeline at one go and then you just it runs and that's give you the final. So it's an iterative process to understand the NGS data sequence analysis, transcriptomics or genomics or anything else. So the next pipeline that takes into account, it is slightly more complicated. We can go to the server and build it right now. So we go to the create a demo pipeline and we click start. We click OK. And then if you see that there are few buttons which are highlighted out here. So Trimomatic, we do the Trimomatic up till pre-processing, it is almost same. And then we do HiSat2. Now, why we do HiSat2? This is what we call combined strategy. The combined strategy is basically mapping onto the genome and as well as mapping onto the junctions. And if you can see, if you want to do individually mapping onto the genome, individually mapping onto the junctions, you can also do that. But that also totally depends upon what you are trying to find. So this we start with HiSat2, which basically combines a mapping onto the genome and isoform, and we call this combined strategy. Now, if we go back to the slide, so this HiSat2 maps it to the junction. Now, what is a junction? What is a junction if I ask you guys? So would you be able to explain to me? If you can unmute yourself or you can write in the chat maybe.
A junction is a joint.
Yes, of course. But what type of joint?
You take the exons or you take the exon junction probably here.
Close, definitely. They're not introns, I guess.
Yeah, they're not introns because we have removed them, right?
Right, we have removed them. Exactly, Sunita. We have removed them. Yes. Anybody else?
Between the two exons?
Yes. Yeah, they are between the two exons, but they are already there. These are the sequences between the two exons, which are connections between the two sequences. Now, if you go to Transcriptomics 1 and Transcriptomics 2 courses out there, the junctions are important. If you also try to understand the junctions between the two exons, that would be really helpful to, these are the steps where you understand how to actually map onto the junctions. Okay, so moving on to the server itself. Could somebody mute themselves, please?
Hello, Velu here.
Velu, yeah, please.
In case here, junctions would mean that we are looking at exons, which are transcripts coming out. We are looking at RNA-seq. So transcripts are coming out from genome.
Mm-hmm.
So it won't actually be differentiating between different exons because they're all spliced together.
Right.
Transcript. So when we look at the read could come out at the junction of exon.
Right.
So in this case, our read could actually be split into two places, and
Right.
where it split, that the algorithm has to figure out.
Right. Right. Exactly. You correctly pointed out the definition. Yes. Please go on.
Yeah. So HiSat2 does that using bow tie. What bow tie does is it matches the reads exactly. What a read matches exactly, it fixes it, and
Mm-hmm.
whichever doesn't have a complete match, it leaves it out. And then HiSat2, the additional algorithm, it breaks it into pieces and tries to match it. And
Right.
if two pieces match at different places, then it is considered as the junction point.
Right. Exactly. Thank you, Velu, for the explanation. It was very necessary. Yeah. So yeah, Velu has told the correct thing. So there are several strategies, as you can see. So RNA, the RNA transcription process is pretty complicated. So while we are getting the data from the RNA transcripts, and we are actually analyzing the data, thereafter, to really understand what are the elements, whether genome elements which have really come in front of you, and what kind of before going to the next level of analysis, it is very, very important to understand that what are the structural, what is the data that we have. So all these data, all this complicated pipeline that you can see, takes care of all the things that Velu has said. So this is what is, takes care of the junctions, it takes care of the isoforms. It takes care of the mapping onto the genome, the known transcripts, the unknown transcripts, the model organisms and the non-model organisms, and building the, updating the GTF file, and so on and so forth is being done. So this pipeline that you can see right now in front of you is, it's a combined strategy, as I was telling you, followed by the isoform construction, a GTF incorporation, mapping onto the transcriptome, and finally building the RSIM table of expression. Okay, so let us complete this thing right away. So HiSat2, and it might be so that your server might become non-responsive if you keep it open for a long time. It's very sensitive, so that's how it should behave. So this is the junction, that's Velu was talking about. So there are pretty good reading stuff here if you can go through it, and also if you go through the topics in the transcriptomics 1 or the transcriptomics 2, you can have a better understanding of the stuff. So cufflinks followed by cuff merge, we update the GTF file here, and then we do the bow tie 2 mapping onto the transcriptome, and thereafter we click, we generate the RSIM table of expressions, and we do the OK, and finally having the results. So so if you click, we do something called, we can do two things out here. One is DSEQ2 and one is N. Let us not do the DSEQ2 right now and click on N here, so that we will explain DSEQ2 once we are towards the end of it, and we have some other things to see. So these are the set of the file that you can see that are generated from this particular run. So the demo pipeline, it can help you to start working on the output of a standard, simple, and also an advanced RNA-seq pipeline right away. So the type of files that are important are these. So you have the exon expression, you have the gene expression files, and isoform expression files, which you can start working right away. So this is how expression table looks like. And expression table is, it being paired end file, it would be fragment per kilobase per million. And if single end, it would be either RPKM or TPM. Now, in case of FPKM or RPKM or TPM, different strategies of quantification are used. Now, gene expression profile. So this is what is a gene expression profile. This is a sample. These are the samples that you can see. This is a particular gene ID, what you can see on the left. This is an ensemble gene ID, and the entire red line highlighted, this is the gene profile of a particular single gene across multiple samples. And we have a number out here. The number which is highlighted right now is 3.27. Now, what is this number? This can be a bunch of different things, you know. The one that you saw right now is actually fragments per kilobase per million. What is FPKM? So the total number of reads mapped onto the genome, divided by, mapped onto the gene divided by the total number of mapped reads in millions and multiplied by the gene length in kilobase. So to give you an idea about the entire thing in a very simple way, because many students are here. And so if you have a bucket of water and you are actually measuring a cup of water, so you have to understand, you know, what is the total water in a bucket, you know, in comparison to the, you know, you have a water in a cup, you have water in a bucket. So if you really try to compare, then you try to bring the cup over to the bucket at the same parlance, at the same proportion. Now, even if somebody comes up where he has a whole cistern of water, so the proportion becomes different, as you can understand. So FPKM, TPM, RPKM, these basically are different types of proportions that come into play, and definitely the strategies of quantifications are different. These are different strategies of quantification of the RNA-seq data. Another thing, how big is the cup? That is also very important. Is it a small cup or it is a bucket-sized cup? So if the gene is bigger, and so statistically more reads could fall onto that gene. And if the gene is short, statistically speaking, again, there would be less noise. So there are different strategies of normalizing the data as well. So you have to normalize something, the data that you have in, you get at the end of this entire pipeline set. Now, and it is known that gene expression data does not follow normal distribution, but it is needed for statistical significance testing. It is very important for normalizing, for transforming the data into some sort where we can use the power of our analysis. So to utilize that power of analysis, we have to transform our data to log normal distribution. And so we transform the scale into the normal scale to a logarithmic scale of 10 or 2, and this would create a good distribution and make our comparison process easier for levels of expression. Okay, so now some amount of statistics which has been there for a very long time, and most of us already know about it maybe. But just quickly going through it. So now we know that how data is produced, and we know how to remove the technical amplifications and preparing the distribution of the data so that we can use this kind of statistical tests, so that so these tests will give you that statistical significance based on the number of observations. So we have 2 groups and a number of samples in each group, and we are comparing multiple observations for that particular condition. The person who came up with the idea for random sampling of observations is William Seeley Gossett, the photo you can find on your left, who worked for a beer factory in US, and to determine whether 2 conditions are statistically significant or not. And this process basically separate between signal versus noise, signal from the data and also noise from the data. So he came up with these formula to understand whether the quality is consistent, and because you worked as a quality assurance in the quality assurance department for a company called Guinness, who manufactured beer, and for beer production, quality of barley is very important. So we take into consideration the mean of the group, the difference between the means of the 2 groups, and also the variance of the 2 groups. Now we take you to the platform once again to really understand what sort of differential gene expression can we do out here. So let us go back to the platform and create something different type of thing. Okay, so create demo pipeline. and we click on start. We click on OK. We do a Trimomatic. We do a PCR clean, and followed by HiSat2, followed by cufflinks, followed by cuff merge, and then bowtie 2T mapping onto the transcriptome, and then produce the RSEM table of expressions. And here is the twist, my friend. The twist is that there is a new box which is highlighted, which is the next immediate step, which is DESeq2. So we have two algorithms in our platform which are called DESeq2 and EDGE-R. So the pipeline actually produces results. We have, let us go to the presentation and find out. So this is the pipeline that I have built right now, and it produces a result like this. Now, what does this result tell me? It tells me that we have a base mean. What is a base mean? We have a base mean of, and we have a log2 fold change, and we have a statistical significance, which is measured by p-value, and we have something called adjusted p-value. So you have to understand the meaning, the mean of that particular gene. Let's say the gene ID out here is ENSG, some amount of 3.13. And this is the mean from the actual expression of that gene. And we have a log2 fold change, which gives you the statistical significance, which tells you whether there was high expression or low expression, and shows you the actual difference between the different expressions that we have. So it's negative, so it might be lesser expression. So something which is positive out here might have, it shows higher expression. Now we also have p-value, and we also have p-adjusted value. It's very important to understand p-adjusted value. So that is the same p-value adjusted by something called false discovery rate. What is a false discovery rate? False discovery rate is the measure is a measurement which is discovered by an Israeli statistician and dealing with microarray data in the 1990s. Now, this is a very important concept, the false discovery rate. So that, FDR was first used in context of microarray, so that you can account for these high throughput observations, which are many. There are thousands, there are millions. T-test, on the other hand, was originally developed for much smaller number of observations. It is more suited for tens or hundreds, and not for thousands or more. So that is why the algorithms that you can see on the right actually uses these high throughput data, these algorithms uses FDR, adjusted p-value, and all these things to really give you that specific quantification of the RNA expressions that you are trying to do here. Now, now that we have all those, all those numbers, we have a lot of numbers out here, right? And now let's try to understand that what these numbers tell us, how this works. The graph in the bar chart, which you can see on the left and on the right, there are two genes across a number of samples in two organisms like mouse and human. Now, if you notice, you would see the number of sample in two organisms under consideration remains the same. The number of samples remain the same. These are the number of samples in the x-axis. Okay, the number of samples remaining the same. But the differential gene expression of the mouse gene, this is the ensemble gene ID, and ENSMUS is the standard identifier of a musculus gene in ensemble. It remains the same. So the number of samples remains the same, but the differential gene expression of the mouse gene is higher than the differential gene expression of the human gene. So these gives us much deeper insight into the functional perspective of genes in a number of conditions in the two organisms that we see. Now let us go to the pipeline and figure out what what files does it produce? So the files have already been already been uploaded out here. So you can see the files here. So this is the set of files which are generated by after doing the DESEC. The most important file that we are going to we would be like to look at is the data set that was uploaded are normal like cancer samples and Claudin low differential cancer sample cell line data, so that we try to understand what is the differential expression of the genes that are there in these two samples. And we have run DESEC. So this is the FPKM value, FPKM value that I was showing you a few minutes back. This is what this file contains. So you might after this lecture you might go back to the server and run the demo pipeline by yourself, and then try to look at the file by yourself. Let us download this file. Okay, let us see what does it contain. How does it look? Okay, okay, and you would like to open it in a spreadsheet program. Okay, and then here you go. Right, so these are the samples, different samples in each of the columns. So how many do we have? We have quite a number of that sample, and these are the genes that are produced out here, which has come out of our analysis. So there are almost 7,000 genes here, 26,000 genes coming out of this. So this is a pretty big number, right? And these are the FPKM values you can see in front of you. You can do some, you can derive graph out of that to really understand what are the genes that are there. You can try to do some scatter plot on your own. We would be covering some scatter plot in the next few lectures, and we'll tell you about how to actually identify based on the results that you have in front of you that you can see right now. We can do some basic machine learning thing like PCA and factor regression to understand more details about how actually the group separates, because it is pretty difficult from this entire length of the table to really understand how the genes have really separated in groups. So based on the values, we try to separate out the different types of cancer that we have. So the different types of samples that we have used. Okay, so let's say we have used TNBs. We have used basal like cancer samples, and we have used Claudin low cancer samples, and we try to look at how on the basis of the values we can separate them out and really try to understand what kind of information does they give it. So there are lots of levels of it. So this is the first level of analysis that you can do with the RNA-seq pipeline that you can have. So please go through the transcriptomics 1 course that is there in the educational platform. Let me show it to you quickly. How can you go out there? So most probably all of you have been able to log on. So this is how the course looks like, right? So this is the curriculum of the course, and so this is something which is a reading material for this, which is associated with the lectures that we are doing. So it tells you about biology of transcription. It is always good to go go back to refresh your basics. So even if you are you might be a very accomplished scientist, definitely we would appreciate that you have joined our program. But at the same time, it is very important to go back to the basics and really study about that stuff, and then get your boots packed in a proper way. And then we quantifying things, and we have interesting quizzes out here, so you can do it. The students would be very interested to that. And then there are logical steps to analyze the data. This actually tells you to download the data on your own, and then maybe you'll be able to analyze the data. So running a lot of demo pipeline would really fortify you, or would really give you that skill set to run your own kind of pipeline. And gene expression table, this would tell you about what is the gene expression table looks like, the table that we saw. So this is best place to start from. Okay. So, and one thing I would request you, after you complete this thing, there is a tab called review. It is very important that we get reviews from you, that because you are our end users, and we have collected all those materials that you can see right now in front of you is basically for you guys. So it's good to write a review. So many people have written their reviews. So these are some of the program participants. So maybe since I have already given my review, so that's why I cannot. So there's a space. So let's say the review box does not appear in my login. So if I go to courses like let's say epigenetics, which I am about to, I was going through it, so I have not written the review out there. So you can find something like this out there. Something like this out there. So see, you can find something like this. So you would like to write something. So it is a good start for transcriptomics or something like this, and then you can write your comment out here, whatever you think about our course. And then please don't forget to star yourself, so that we would like to have a starry review from you. But if you think that if we deserve less stars, you can are also definitely welcome to do it. So so much for today, and this is how our review looks like. And if you on the next day we are going to cover something about filtering, removing noise, and normalization techniques, which is connected to the RNA-seq data expression, data analysis. We'll be looking at some amount of PCA visualization to really separate out samples. PCA is the basic step of dimensionality reduction, because right now we saw almost 26,000 genes. What are you going to do with that? I mean, there are a huge number of genes and really difficult to find out the expression from the right kind of information from there. So we have to reduce the dimensionality and try to figure out what exactly they mean. And we would look at something called principal components, and obviously variance within the data, which are the outliers, which are the filtering techniques, and some normalization. So I would like to take the QA. Before that, I would like to tell you that we have recently launched a program called Biomedical Data Simplified, Biomedical Data Science for Biomedical Data Science. And one of our faculties out here with us right now, I would like to give the stage to Mohit, Dr. Mohit Mazumdar, if he can tell little bits and pieces about the information that he has about this course. Mohit, all yours.
Yeah, thank you, Pratham. Yeah, I mean, we'll be launching this program called Biomedical Data Simplified. So where we will be talking more about data science aspects of biomedical data, how we, make meaningful understanding out of data, this big data, using machine learning and data science techniques. So we had the first webinar yesterday, very successful webinar, where many people joined and they learned about the courses, and also we had a small demonstration about PCA using classification of the cancer cell lines that Pratham today talked about. So I think we will also be sharing. We have been sharing the video to all the participants of different programs. So we have our Facebook page and our LinkedIn page, and also you can follow us in Instagram as well. So I mean, we've been keep posting on those platforms about the free webinars that we've been doing. So everyone is welcome to join those free webinars to learn more. So we'll be doing another webinar on 10th of September, the second of the sort. So there we will be discussing more advanced techniques about data science and machine learning. And in the next after that on 16th of September we will be launching this program. So thank you, Pratham, and that's the information that we have right now.
Yeah. So on September 10th, there is this free webinar once again. So if you're interested, you can write to us. You can follow our LinkedIn and Facebook profiles, and also LinkedIn, and also Twitter and Instagram. We are regularly posting about these programs later on every day, and then it would be a good learning for you, I guess. So any questions you have, I would be happy to take. We don't have much time left for today's session, only 5 minutes. So if you can go ahead and ask me. Let us look at the chat. If we have something here. Okay, so yeah. So we have yes, and can I have the video link of the biomedical data simplified? Yes, Shobhadeep, I think you have been sent an email about about the thing, about this biomedical data thing. So it is going to come up again on September 10th. You can attend it. So to know about the kind of things that we are going to cover, and obviously we can share the video link of yesterday's session as well. Log scale, yes, Naresh. What do you want to know about log scale? You can unmute yourself and tell me if you want.
Why we need to transform this linear values to the log values?
Yeah, so I was telling you about the gene expression data does not really follow any normal distribution, but any statistical process should always start with some distribution which is always has to be normalized. So that is why for normalizing the thing, we use log scale transformation to the base 2 or to the base 10, so that the subsequent statistical tests we use to derive the mean and to derive the median, and also the PCAs and all those machine learning principles that we use to analyze the data, to find insights from the data, it is very important to transform that into the log scale. That is the whole reason. Does that answer your question, Naresh? Okay, thank you very much. So Shomodeep, you haven't received an email, or yeah, you haven't lost it. So usually the emails which go around for, so I would like to reiterate the fact that most of the emails that we send about the class, about the previous day's lecture and also the upcoming class, the Zoom link, etc., usually gets deposited in your in your update tab. If you're using Gmail, it would be in your update tab or in your promotion tab. So I would request you to look into the promotion tab or in the update tab of your Gmail. You can get it out there itself. So that is the whole thing I wanted to mention about it. What is a microarray? Microarray is basically some amount of it is again a high throughput sequencing procedure. And yes, microarray data are a little noisy because it contains a lot of artifacts from the machine which needs to be removed. There are signals out there which needs to be removed out there, and microarray is definitely the first process, the first onset of high throughput data and high throughput generation of the data started with microarray. So you can read more about it, maybe in the web, maybe.
Does anybody have any questions? Sorry if I intervene. Anyone? Okay, so I just wanted to take the stage and tell everyone about how to solve the issues of logging in to the Zoom meetings. For the future, if you are unable to log in, there is the meeting ID at the last.
Sorry, do you want to share your screen? That would be more, I think, more apt for the people to understand. If you want to share your screen and tell them.
Just a second. Okay. I don't think I would be able to share the screen and show about the login procedure, but let's try. Okay, wait. So can everyone see the screen?
Yeah, we can see it.
Yeah, okay. So to log into the Zoom meetings, just copy the link here. And once you try to set enter, you'll get this Zoom sign-in request. So go to join a meeting and copy the last section of this Zoom meeting ID. This is the Zoom meeting ID. Paste it here and join. That would not ask for any password or any hassle or hurdle in between this. We are trying to refine this process of not going through all these hurdles. But for the time being, if you are unable to log in, just copy this ID. This is the meeting ID. Copy it, go to join a meeting and paste it out here. Okay, I hope this helps everyone. And if you face any issues, you can type us in the chat box or in WhatsApp and even on the mails. Thank you.
And
Okay.
I think if you install the app in your mobile or in your computers, then this problem will not happen.
Yeah, so you can also download the application for Zoom meetings, and thus you will be able to log in to the Zoom via your mobile phones and anywhere from any else. Okay.
Thank you, Apar.
Yeah, thank you, Apart. Thank you, Mohit, for intervening. Yes, that's nice. So all of our classes takes place in Zoom, and all of our webinars also takes place in Zoom. You better, kind of get a hang of this process and try to join our meeting accordingly. Okay, so much so, I think we are done for today. And then thank you for joining us. See you on the next class.